This is the Kratom Science Journal Club with Dr. Jonathan Cachat, neuroscientist and expert in psychopharmacology. In each episode, we discuss an article in a peer-reviewed journal. I'm your host, Brian Gallagher, blog and social media writer for KratomScience.com, your source for all things Kratom. Name of the study: Effects of Nutrient Fertility on Growth and Alkaloidal Content in Mitragyna speciosa, Kratom. And this is the Christopher McCurdy team down at University of Florida who've done a lot of Kratom studies, and they got the NIDA grant money a couple years ago. I'm not sure if the study is from that, but so this is unlike one we've done before. They're actually growing kratom trees, and they are testing it for alkaloidal content. It says 68 kratom trees were vegetatively propagated from a single mother stock to reduce genetic variability and subjected to four varying fertilizer application rates. Leaves were analyzed for chlorophyll concentration, biomass, alkaloidal content to understand the physiological response of the plant. So,
indeed, yeah. So the first study that we've looked at, I think the first study that I'm aware of is published that looks at the various influences of the cultivation environment on the alkaloid content in kratom. So it could be of particular interest to anybody who is growing kratom and they want to know what the ideal sort of nutrient range is or not is, or maybe they don't get that informed too much at all. But I thought there were some interesting takeaways overall.
So they basically use Scott's fertilizer at they use the same fertilizer at four different levels. 15, 19,
0.
12, if you want to know the NPK.
Oh, yeah. So the NPK, what was the NPK?
15, 9, 12. That's a Scott's fertilizer. And
the different groups, so essentially of these 68 trees, they were in 11.4 liter containers, and they were given four different nutrient treatment groups. So 0 grams of nutrient, 46 grams, 74 grams, and 96 grams of nutrients.
Yeah.
It ends up being somewhere between, like, given the size of the container, it ends up being somewhere like around 6.5 and 8.5 grams per liter of the cultivation container.
Yeah, and they're watered a lot. But kratom grows in kind of swampy areas. One of their main problems was that there's not a lot about kratom production anywhere in any kind of literature. So they kind of just had to experiment a little bit. And they used all plants were grown under natural daylight, irrigated twice a day with approximately 2 liters of water twice a day, which sounds like a lot. Greenhouse heaters and fans were controlled by an environmental control system. So had you ever done anything like this with cannabis? I know you studied it a lot. Yeah.
I mean, we did just this, and it's essentially, the first steps towards optimizing growth for commercial environments. And, so in the scheme of things, they're really just looking at growing inside of a controlled environment and only modifying the nutrient content to just get a very broad indicator of whether or not you can increase the alkaloids based on the total amount of nutrients. It's a little bit different in cannabis because we have some baseline information about its cultivation. So whether you're looking at light, temperature, humidity, nutrient content, or any other sort of additives, there's much more sort of laid down in a foundation for cannabis. But you need studies like this to at least get the ball rolling for kratom.
Yeah, the thing about alkaloids in kratom or in any plant is they serve a different function for the plant. other than just to make us feel good. It even says, although alkaloids can serve to benefit human health, their primary purpose for being produced by plants is to support important ecological functions such as defense of pests and disease. I think maybe tobacco I've heard that with, where the nicotine actually kills aphids or flea beetles or some kind of pest that would otherwise eat the plant.
Right. Yeah. So these plants aren't biosynthesizing these compounds in order for our enjoyment, and at least we can't force them to until we understand this. And I think one of the elements that was somewhat missing from this paper was the direct link between like input nutrients and biosynthesis of the alkaloids. It wasn't like, well, we know that in the biosynthesis pathway, way upstream, there's precursor A, and precursor A is derived from nutrient set, A2. And so by adding more of these, we should push the biosynthesis over the edge. I don't think that pathway, that biosynthesis pathway, is that fully described. So it was something that was somewhat missing from here, but I don't think that we know that yet. And making a bigger connection between nutrient concentration and alkaloid content would be the first step in sort of understanding those physiological pathways, those biosynthesis pathways.
Is that kind of where testing the amount of chlorophyll comes in? Because I guess that would measure the health of the plant against the amount of alkaloids in it. Is that the idea there?
I think it. Yeah, so I mean, the more chlorophyll activity that would be in the plant, the idea would be that, like, the machinery, the biosynthesis machinery would be engaged to a higher degree or higher rate. I think the point in this paper was really, I mean, so they're looking at the biomass, and they're looking at, tree height, width, trunk growth. So the overall biomass of the plants as a function of the nutrient content, the chlorophyll, alkaloid content, and then they confirmed all those. Like, so essentially they did an extraction procedure that was an ethanol wash where things were sonicated and then evaporated, and then they quantified them with an HPLC. So I don't know if I necessarily saw a direct correlation between chlorophyll and alkaloid content, other than just that if there's more plant mass and more plant activity, that would indicate maybe potentially some greater concentrations of the alkaloids. But that's not what they ended up finding.
Yeah, I mean, I know they definitely, it's like the more fertilizer they used, the bigger the plant was, and the more actual leaf material they had, which is kind of, you'd think that.
And by default, therefore, that they had this more alkaloid content. So there was more fertilizer equals more biomass. Fertilizer to alkaloid relationship was not linear. It was highly variable. So what was interesting is that fertilizer rate had little influence on the concentration of mitragyna. They looked at 1, 2, 3, 4, 5, 6, 7, 8. Or is it 8? 1, 2, 3, 4, 5, 6, 7. Yeah, 8 of the different alkaloids. And basically 4 of the alkaloids had no change between high and low fertilizer, and that included mitragyna. So there was really no, if we increase the fertilizer, we increase the mitragyna content. They found no change there. 2 alkaloids where there was high fertilizer, high alkaloid, and 2 alkaloids where there was low fertilizer, low alkaloid, which was pretty interesting in the scheme of things, especially if you're trying to develop some sort of commercial profile of kratom alkaloids.
7-hydroxymitragynine was below the lower limit of quantification in all samples throughout the study. Mitragynine was detected in 43% of the sample leaves, but was below the lower limit of quantification at the beginning of the experiment and was not influenced by fertilization. That's what we were just talking about. And they also said somewhere else that the amounts of mitragynine were lower than in commercial samples. So that kind of, like what I've heard with Josh, Kratom Science Josh told me was that he was told that it takes several years to really develop the alkaloids, kind of like wine grapes take a few years to grow before they're good enough to make wine out of. And he's told it takes up to 7 to 8 years, and you basically need to stress the plant for it to produce. by picking leaves in low water. Supposedly, picking the leaves will make it produce more as a defense mechanism to prevent the leaves from being eaten. So it's kind of like I don't know how long this experiment was. It seemed like it only went over the course of a season until they got full-grown plants, and it might not have...
So there was 12 weeks, 87 days total once they were planted in that larger, the larger 11.4 liter pot.
Okay.
And you're right, that did the comparison to the concentrations of commercial. So I looked into that a little bit more. So it says Mitragyna not influenced by nutrient concentrations, and also this study's plants, Mitragyna levels were 4 to 5-fold lower than those determined in commercial products. So commercial products would measure at like 1 to 6% Mitragyna, per weight.
Yeah.
Whereas this study, it didn't even get, half of that. It was 0.5%, not even 0.5%.
Yeah.
And other studies, they mentioned citing like 12 to 66% of the weight of leaves was Mitragyna of trees collected in Malaysia and Thailand. And I have heard that as well, that the age of the tree, and especially like being over 2 to 3 years old, is really when the alkaloid content starts to kick in it's in a significant way, where you're seeing these 1 to 6% Mitragyna. So it was a relatively short lifespan, and they were brand new plants from the beginning. It's, it would be interesting, and I hope that this group has plants that are continuing to grow from this original study, so they can start looking at production.
Yeah. I don't see why they wouldn't. If they already have the greenhouse set up, unless they need it for something else. But yeah. Hopefully, it turns into a more long-term thing. But what did it say about the alkaloids here? It said, in general, fertilization did not affect the average painanthine, speciosiliantine, mitraphyline, and corinoxine concentration per dry leaf mass. And but if we decide to do that other alkaloid paper next time, I'll work on the pronunciation of all of these. But it said that wasn't affected by fertilizer, those particular alkaloids. It says, given no consistent temporal trends were observed for alkaloids quantified in this study, data was pulled together to increase the statistical power for analysis of overall alkaloidal trends. What does that mean exactly?
So you're asking what they pulled together, how they actually pulled that together?
Yeah, I'm not exactly sure what that means.
I think essentially what they did was they pulled together the days. So in table 1, we have this, week 0, week 2, week 3, week 4, week 5, all the way up to week 12. And I think essentially what they're saying is that they pulled together all of those, the entire temporal, they collapsed it into then table 2, just the average alkaloid concentration. And then that's where we got that 4 alkaloids. There was no change between the high and low fertilizer treatments. There were 2, the M and the C, that were high fertilizer, higher alkaloids, and then the iso and the corthinoxanine were lower. So lower fertilizer, higher alkaloids. And you can sort of see that play out in the data where there are higher numbers. The first 4 are essentially the same between control, low, medium, and high. The 2 after that are higher as we go down, and then the last ones are higher as we stay up at the top. It gets smaller as we go down. So that's what they mean by they pooled together. They didn't necessarily see a temporal characteristic. A number of the alkaloids were below the limit of quantification all the way up through at least week 6. Before that, they couldn't even really detect Mitragyna.
Yeah.
Once we got to that 6 week and beyond, they started detecting a detectable, a quantifiable amount of Mitragyna, but it was still, pretty low relative to what's reported in the commercial samples.
And they had some other things in the discussion to say about Mitragyna. Cross-section survey of Malaysian kratom users indicated that medical efficacy of kratom tea consumption was maximized when consumed within 24 hours of brewing. Effectiveness of kratom tea was reported to decrease 48 to 72 hours. However, laboratory analysis of the kratom tea they're talking about in this study they're referring to found no difference in mitragyny concentrations. So results
over
suggest,
that time.
Yeah, over 48 to 72 hours after brewing, but they said the effects weren't as good. And it says, thus results suggest changes in compounds other than mitragyny might play a much more significant role in reported medical effectiveness of kratom.
That part's real interesting because essentially
Yeah.
what they're describing is an entourage effect. To say that just the amount of mitragyny doesn't necessarily result in reported human effects, and that if the level of mitragyny is staying constant, but over these 72 hours, some other smaller, less known alkaloids are degrading or changing their concentrations, that can be the cause or the result of why people are reporting them differently. Like it's mitragyny plus 3 other alkaloids in some sort of entourage effect. That's very interesting.
Yeah, and they even said a related study examining mitragyny presence in leaves collected in the Philippines only detected it in 38% of collected samples. Get to look at that study because I don't know if people were consuming the ones without mitragyny and reporting effects, but that was pretty interesting in the Philippines.
So just to I wrote up a summary here just of the if you were to be growing kratom trees and you wanted to know, based on this research data, what would be the best amount of nutrients for your plant, basically because the more fertilizer equaled more plant biomass, and then essentially therefore the overall alkaloid content, you can recommend that if you have, it's between 75 and 100 grams of nutrients per 11.4 liter pot, or 6.5 to 8.5 grams per liter of whatever pot that you're growing in. And what that achieves is not necessarily a higher alkaloid content, but higher plant biomass. And they noticed a significant difference between no nutrients and then nutrients in terms of overall plant biomass.
Yeah.
But then between the low, medium, and high nutrient schedules, there was no significant difference in that plant biomass. So not using nutrients is not a good idea. Using a reasonable, sort of mid-range amount of nutrients is what the target is, essentially. You don't need to go too high, you don't need to go too low, but using them definitely makes a difference overall.
And also, I mean, they were growing. What I thought was funny is they're in Florida and they were growing these in heated greenhouses too. So I guess depending on what time of year they were doing it, maybe they had to start in the winter. I mean, I've talked to a guy on this podcast that actually grows them, and he's in Ohio, and he keeps them outside in the summer, and he just brings them in and puts them under probably fluorescence in the winter. So I don't think it necessarily has to be a tropical environment in order to grow them, but I don't think you could keep them out in the winter though.
Yeah, I mean, unless you're in Florida or Texas, you see a bunch of guys on Reddit in Florida and Texas who get the benefit of keeping them outside all the time, maybe even putting them in the ground out there, which I think is good, in relation to the longevity, right? So keeping them for years, in order to get that alkaloid content up. But yeah, you can certainly recreate the greenhouse environment. I think that they were around 74 degrees Fahrenheit for our non-metric audience, and the daily light integral that they had was pretty normal. Like there wasn't an exorbitant amount of light needed. Like generally in cannabis cultivation, that the amount of light will be 1.5 to 2 times the daily light integral for normal plant cultivation. They're trying to increase that light. So yeah, I mean it can certainly be done. I think the other cool angle or element of this was that they very clearly laid out their alkaloid extraction, and it doesn't require any really sophisticated technology. So they just took their leaf and ground it up. So then they just, they powdered it up into the powder, like you could get a powder from a smoke shop. They put that into the ethanol, the 190 proof ethanol, and then place that into a sonicator. A sonicator is like essentially a water bath that you would clean jewelry in, but it just is, it vibrates very quickly.
Yeah.
So after that, they separated them out. They separated the ethanol and they separated the layers. They used a rotary evaporator. You wouldn't necessarily need one of those, but they did 3 washes with this ethanol. And then at that point, they had essentially a concentrated alkaloid solution that they were able to analyze with the HPLC technology. So, it's just a regular ethanol wash. It's pretty well described. That could be done. Don't necessarily need expensive laboratory equipment to get it done.
See, I'm wondering why they didn't maybe try to get information on production practices, like maybe from Indonesian farmers. And it even said Kratom production practices are largely undocumented and thus confound application of established management practice that aim to maximize plant growth and alkaloid synthesis. Information on relationships between horticultural practices and plant forms is limited, but crucial to support development of newly emerging Kratom production industry. So essentially, we're kind of like in the dirtweed, dark ages of Kratom production. You get good stuff from Thailand. That's how it used to be with cannabis.
The commercial production has been coming from leaves that are essentially just growing wild.
Yeah.
I mean, there are farms over in these countries where like the trees will be lined up in rows, right? So like that's the extent of the commercial production. They're just old, established, existing trees that they pull from. And I mean, I've seen pictures of some of these trees where they're 30, 40, 50 feet tall. Like they're just enormous. So I would say that like really commercial production, specifically for the purpose of getting raw material for commercial Kratom products is literally just like you're saying at the very beginning. And before there are no like best management practices because before now they were just grown in the wild.
And it seems to be, they probably don't even use a lot of fertilizer or any inputs or anything like that. Just seems like just from the YouTube videos and stuff I watched, they're basically collecting it, and and I mean there's farms with rows of trees and stuff, but they're basically going out collecting the leaves and drying them out.
And these plants are, years old. We count their age in years, not in weeks, which they did in this study, and they had to start somewhere. But I think you're right. Hopefully we can assume that the group in Florida has this greenhouse going and they're going to keep it going. And why not do something on the time scale of weeks just to see? And now they essentially know too where the optimal, should they treat with nutrients, yes or no? Yes. And they have an optimal range to increase biomass and plant health. So hopefully now they can continue that over time. And then, what we're going to see, like in cannabis, you would add things like simple sugars and other additives. I don't want to say necessarily nutrients because it's not the NPK sort of set of elements or nutrients or minerals that we're looking for. It's beyond that. So like simple sugars or carbohydrates or something. Is there other elements and additive things that can be added to the soil amendments, if you will, that will then actually modify this alkaloid content? Or what happens as a plant goes from like weeks to months to years old to where it starts switching and really kicking in this production of the alkaloid content to where it's, between 1 to 10% or so per leaf mass. I don't think we know that yet. And so this type of line of research done by the group down in Florida is really the first to peer into it in any sort of empirical way. It's one thing to sort of pull the leaves off the plant and brew a tea and say, oh, it's getting there, right? And we humans have the ability to detect that. Those thresholds are observable and noticeable to us, but it doesn't put a hard quantification on the number. So, it would just be kind of weird if you're looking at it, 52 weeks in a year, let's say you get to like week 75, and all of a sudden the alkaloid Mitragyna alkaloid content goes from less than a 1% to 7% in 1 week. Like, what happened during that week? That's really what we want to understand
Yeah.
if we're going to try to grow for commercial purposes.
Yeah. And it also said something about the kratom trees utilized in the study were either kratom trees utilized in the study were likely of a different genotype than those used as a source for commercial kratom products, or another key environmental factor responsible for mitragyny and other alkaloids was not present in our study. So what is that about genotype? Would that be like the equivalent of like hemp used for fiber to cannabis used for smoking?
That would be genotypical, but I think really when you're looking at the different strains, so what makes a Blue Dream different from a Tangy is not really entirely genetic. I mean, there's some genetic material there, but it's not. You can't distinguish them as different species. Although I actually just heard about a study out of Canada that this guy's been looking at cannabis genotypes for like decades now, and he finally has just produced a test that can tell whether or not a plant in its infancy stage is going to produce more CBD versus THC. So that'd be between hemp and between cannabis or medical cannabis. But so in this study, they say plant cuttings were taken from a single mother stock obtained from a private plant producer to reduce potential plant genetic diversity among replicants. And so it's sort of pros and cons to that. Like, of course, they would want to keep all the plants and the genetic stock of the plants the same, because really what they're looking at is a difference in nutrient content. Mm-hmm. If there was genetic variability in there, it would be confounding variable, because the influence of genetics with nutrients would come into play.
Yeah.
But by just saying that it's a private plant producer and not really getting into the sort of provenance or history of where those genetics came from, it's hard to say whether we're running into an issue where, like, oh, well, in Thailand and Malaysia, where they have these higher alkaloid content trees, it's probably a different strain or a different genetic type. We don't know that. And it doesn't seem like the authors of the study were concerned. I feel like they would mention that to say, like, this came from a stock that originated in Malaysia, but has been grown in the US in Florida for X amount of years. There's really not a lot of details on the provenance there to lay things out. But, they had to have that genetics similarity across the board. They could redo this study almost exactly the same and just use different genetics rather than modifying the nutrients. And then, then we could see, but I don't know what sort of access they have to with this private plant producer. And what sort of different varieties or genotypes they may have. Without the provenance there, like, it certainly wasn't a commercial kratom farm in Florida that's like getting ready to pump all this stuff out. At least, or maybe it was. I don't know, but I don't think so. I think it was more of a hobbyist type thing that's now maybe gearing up or pointing towards commercialization.
This is just a guess, but it seems like it would have to be grown in somewhere like Florida. I mean, I guess they could be greenhouse grown, but it seems like if the trees have to be a certain age, then they have to be a certain height, and you'd have to get some pretty high greenhouses to grow them in. But you could probably train them to be wider rather than higher.
Yeah, I wouldn't be. Age is not limited by height. You could definitely train it and like keep them light. You could bonsai the thing, and you could pretty old and not a lot of space. So I would be surprised if this group is not on the path to doing that. We'll just have to keep, as with almost all kratom studies. This one was published in December of 2020, so less than a month old, and we try to find the papers that are coming hot right off the presses. So we're keeping everybody at the cutting edge. But this is where we're at, and this is, as far as I'm aware of, the first empirical paper that looked at these growing conditions as it relates to alkaloid content,
especially on like the volatile nature of how these things are imported from Indonesia. There's, an FDA import alert. Indonesia's government keeps talking about outlawing it. But then in Thailand, they just legalized it for medicinal purposes. And I think they just completely legalized it this year, but it seems to be going slow as to how that's going to pan out and whether there's going to be a commercial source from there. And and another thing is you have to get the plants to grow it. It's like really impossible to grow from seed because like they germinate at a very low rate. I've heard that. That's another.
I haven't heard about anyone starting them from seed.
Yeah.
And I just did a Google search for American Kratom farm, and really there's nothing that immediately jumps out. There's stuff from the AKA, but it's more about good manufacturing practices, less about the U.S. I think I've heard of some groups over in Arizona too. So I would be surprised, given the risk potential of like shutting down import of raw kratom, that there aren't at least one or two groups in the U.S. that are getting a farm ready, just in case that were to happen.
Yeah, really. Yeah, I mean, it would be a definitely good thing to do if you're a farmer and you can figure out from the not very much information how to do it.
Well, and really at this point, it seems like you're on the right track if you get the plants in the ground, and that's it. You just got to keep them alive for years.
yeah. Just get them in the ground, test them, and see what happens.
Get them in the ground, keep them alive, and hopefully it doesn't snow or rain.
Well, yeah,
or freezing rain. I think the other interesting thing to sort of walk away from this paper with is the notion that we talked about earlier of the entourage effect, and that often we'll get a lot of questions sent in to us about, we're sick of hearing about Mitragyna and 7-hydroxymitragyne. Tell us about the other ones. And there have been little study on like, highly concentrated other alkaloids, the speciosa, the one that starts with a S.
Yeah.
It's hard to get that in like high enough concentrations to where you could have a human just take that and see how that works out. What they're saying though is that the mitragyna content alone doesn't seem to affect the reported effects in humans. So with the T there. And so this notion that they're working together in concert and there's some sort of entourage effect is, I think, pretty compelling. And as we elucidate that more, maybe it's that like sort of the sociability and antidepressant effects are mitragyna plus alkaloid 1 and 2, and the pain relieving is like mitragyna plus 3 and 4 in different concentrations. And that would sort of be like where you hear in cannabis people trying to make, certain ratios of cannabinoids to treat certain disease subsets or abnormalities. Mm-hmm. Maybe we're heading that way in kratom. And if we get another plant that has this entourage effect, it would be pretty interesting to see the similarities and overlap between the cannabis and kratom.
Mitragyna alone might not even be a good synthesized or
good target, right?
Yeah,
right.
Good medicine. Because I think even extracts aren't mitragyna alone extracts when you buy them from a company. They're alkaloid extracts, so you still got the alkaloids working together.
There are some where it'll say, pure mitragyna isolate. And based on this notion that there could be an entourage effect, those isolates aren't necessarily the goal. You want to have an alkaloid content that's a mix of all of these alkaloids, not just one.
So if you know anything about kratom cultivation and alkaloids or any of the topics we covered, if we didn't cover them enough, we got something wrong, leave us a comment. Check us out on Twitter at Kratom Science or at Cachat. Dr. John can also be found on ccvresearch.com. The song is called Moonrunner. The band is Captain Big Wheel. The Kratom Science podcast, Kratom Science Journal Club, is produced by me, Brian Gallagher, for KratomScience.com. Take care.